Good morning, judges. My name is Sohan Karidi, and my project is in silico novel inhibitor for dilated cardiomyopathy, DCM, through computational phytochemistry in the medicine and health category. In silico means scientific experiments or research conducted or produced using computer modeling or computer simulation. And computational phytochemistry uses chemical extracts from plants to find a non-toxic candidate drug for DCM. What is dilated cardiomyopathy? Dilated cardiomyopathy, DCM, is a highly prominent disease affecting 1 in 500 people. When a person has DCM, their heart overworks itself, thus causing the heart structure to dilate/distend and increase heart rate and blood pressure. The current drugs target suppressing blood pressure, heart rate, or both, and target other organs. The main DCM drugs in the market go by the acronym A, B, C, D, and D. These stand for ACE inhibitors, beta blockers, calcium channel, digoxin, and diuretics. Although these drugs are beneficial, they give the patient many side effects from lung, liver, and kidney failure to cold, headache, vomiting, etc. Finding a non-toxic drug targeting only DCM is a key to improved care. My hypothesis states that if the function of Beta-1 Adrenergic receptor, ADRB1, that regulates heart rate and myocardial contractility is treated with naturally occurring phytochemicals, then dilated cardiomyopathy can be alleviated with low toxicity because phytochemicals are natural and offer lower toxicity than synthetic chemicals. How do drugs target the affected cells? Since the project's scope is to create a non-toxic drug for DCM, finding out how drugs specifically target infected cells is crucial background research. Since drugs target cells, I first started researching the cell. The cell is filled with organelles, and around the cell, there is something called the cell membrane which all incoming/leaving material must pass through. Zooming into the cell membrane, we can see a wide array of proteins, including a protein channel which all material must pass through to enter the cell. The protein channel is a two-step mechanism comprised of a receptor and ligand. The receptor is the lock, and the ligand is a specific key that unlocks a particular lock. From this research, I now needed to know which cell holds DCM, and how do I unlock that cell. In this process, I have used a complete quantitative methodology. But what are quantitative methods? A quantitative methodology is a methodology that objectively analyzes large amounts of data to draw statistically accurate conclusions based on the hypothesis. The project is 23 steps, however, these steps can be further subcategorized into 4 main color-coded categories: ADBR1, pre-existing drugs, plants that ameliorate, and HTS application. Now that I had isolated the root cause of DCM, I needed to extravagate the independent and control variable from this. Through using the protein database, I was able to find the molecular format of ADBR1. The molecular format was in star.pdb format, and the molecular docking program used in the future require the ADBR1 molecule in star.pdbqt, simulation-ready file format. Using a program called Autodoc Tools, I converted the file to *pdbqt format. As you can see, has many differences from star.pdb format. The model in star.pdbqt format is now my independent variable. Since ADBR1 is a receptor/lock, I needed to figure out where the ligand/key goes. This site is called the binding site. Through researching, I could not find the 3D coordinates of the binding site online, as the area of study for ADBR1 is generally tiny. Reflecting on the model downloaded from the protein database, I realized that it was already bound to a small molecule called ALE1401. Since ADBR1 was already attached to a molecule, we could find the binding site if we could figure out where ADBR1 was bound. Using Autodoc Vena, I opened the amino acid sequence, and then found an isolated ALE1401. Following suit, a 3D grid box was generated, moved over ALE1401, and made the smallest size possible to reduce variability. The coordinates of the binding site will serve as the control variable for the following steps. Before I started my research into making a DCM drug, I first wanted to see how effective the current DCM drugs are. I could only find 3 drugs with their chemical structures online through research. Using the Zinc database, which houses over 980 million different chemicals, I downloaded these drugs into instar.pdf format. The molecular docking program needs all files to be in star.pdf format, so using a program called OpenBabel, I converted all the files to star.pdbqt format. The following input required by the molecular docking program was a configuration file. A configuration file houses the independent, dependent, and control variables. The molecular docking program used in this project is Autodoc Vena. Autodoc Vena takes in the inputs and displays the output in an affinity value in kcal/mol, where the lowest value is the most potent drug. The independent variable was the ADBR1 molecule, the dependent variables were the 3 drugs found online, and the control variable was the binding site coordinates and 2 more values. The energy_range value is a program default value, and the exhaustiveness value tells the program how many times to run the simulation and displays the average results. To reduce any variability, I set this value to 10. Keeping in mind that the lowest value is the most effective, we can see that digoxin with an affinity value of -9.5 kcal/mol looks the most effective. However, to get an overall sense of the data, I average all the values to -7.8 kcal/mol as a baseline comparison value. To take a step forward, I first took a step back. Rather than finding the exact phytochemicals, non-toxic chemical extracts derived from plants, I first researched and found 13 plants related to DCM or heart disease in general. From these 13 plants, I further researched the specifically non-toxic phytochemicals of these plants and comprised a list of 187 phytochemicals. Following a similar set of procedures, I first downloaded all 187 files into a star.sdf file. Then I converted all of them to star.pdbqt file using OpenBabel. Then a configuration file was prepared for each file, and simulations were run using Autodoc Vena. The independent variable was the ADBR1 molecule, the dependent variable was the 187 different phytochemicals used, and the control variable was the binding site coordinates, the default energy_range value, and the exhaustiveness value. The average results chart shows that Hawthorne berry and Terminalia arjuna L. offer the most promising plants for a DCM drug. Taking a quantitative look at the data and observing each plant's high, low, and average value, Rose's plant has the lowest weight of -10.1 kcal/mol. This value is not only the lowest, but it also is 3 fundamental values lower than the pre-existing drug's affinity value baseline of -7.784 kcal/mol. High-throughput screening, HTS, is an application used to screen through millions of non-toxic chemicals in a short period to find a potential drug. Located in Austin, Texas, an HTS application with a non-toxic chemical library of 3.18 million, split into 5 categories. The HTS application has a similar user interface to Autodoc Vena, where the independent, dependent, and control variables are required as input. The output of HTS is the top 1,000 results in each of the 5 categories. The independent variable was the ADBR1 molecule, the dependent variable was the 3.18 million non-toxic chemicals screened, and the control variable was the binding site of the ADBR1 molecule. The average affinity values show each category around -9 to -8 kcal/mol. A quantitatively look into the data and seeing the high, low, and average values in each category reveals the actual results. Keeping in mind that the lowest value shows the most potential for a trial drug, and that the baseline comparison value was -7.784 kcal/mol, we observe two values of -12.1 kcal/mol, which show phenomenal results. The graph shows that the pre-existing drug digoxin seems to be the most effective for the average affinity values. However, the actual results are shown when looking at each category's high, low, and average age values. As we can see in the plant category, the plant rose has the lowest affinity value of -10.1 kcal/mol. In the HTS category, we have two sections with an affinity value of -12.1 kcal/mol, which shows the most promising chemicals for a DCM drug. Furthermore, in the plant category, 22 phytochemicals were observed to have an affinity value lower than the baseline comparison value. Over 2,000 chemicals in the HTS category were honored to have values lower than the baseline comparison value. Looking at the 22 phytochemicals in the rose plant, we can see a massive variation, with some values low and others high. The lowest value of -10.1 kcal/mol comes from the phytochemical B-carotene. French zoologist Jean-Baptiste Lamarck invented the Lamarckian genetic algorithm. It follows a series of mathematical formulas, as seen in the green. The Lamarckian genetic algorithm is the most widely used algorithm used in drug discovery, as it offers more functionality than the Darwin or Baldwin method. In conclusion, based on the results, we can claim that our initial hypothesis is statistically accurate through silico analysis in finding a naturally occurring phytochemical with low toxicity that can ameliorate dilated cardiomyopathy. As shared in this project, through in silico analysis using pre-existing drugs as a baseline comparison value, a phytochemical, B-carotene, from the rose plant and chemical, Z29689317774_1, from the HTS category and amine PC have been proven to have an affinity value significantly lower. Armed with a drug discovery that ameliorates dilated cardiomyopathy, I would like to partner with a pharmaceutical company to conduct preclinical trials and clinical trials and receive FDA approval to improve human life one rose at a time.